again. Nice to see you all waiting here for me. What I'm going to do is talk to you a little bit about some work that I started doing with Tom when I first began as a postdoc in his lab, so right after my PhD. And I continue today, really, especially in the first few years that I was at Wellesley. And it's addressing this question of how a well-mated queen can help a colony become busier, to help produce harder working, faster moving, more active bees. So you heard a little bit yesterday, if you saw Kim's talk, about some of the ins and outs of the sex life of the honeybee. But if you didn't, you probably know those juicy details anyway. I will remind you that virgin queens mate one or several times during the first week of their life, but that is really the only time they're going to go out and harvest semen from the environment. They travel to these drone congregation areas, shorthanded as DCAs, and there they are met by an eager crowd of drones who have been waiting for one of them to show up. They immediately orient to the queen, they chase her through the air, they form what's known as a mating comet behind her, and really, the queen, people often ask me how the queen chooses who she will mate with, and it's not really a choice so much as the fastest man wins, but that's a good choice in terms of natural selection because it probably means that fast male is healthy, vigorous, came from a well-nourished colony, and is ready to pass his good genes on to her future progeny. So the fastest drone wins, and in scientific terms, we call this extreme polyandry, this kind of mating system where a single female mates with many, many, many males. And the question is, just how many males? In terms of social insects, this number can be huge. When we look at the number, and I'll get to that in just a second, but when we look at the number of social insects out there in the world now that we know to have this kind of extremely polyandrous mating system, it's vanishingly small. So we know about 40,000 species of social insects that have been counted for, and of those that have been investigated, we've found extreme polyandry in just over 30 of them. So that's about 0.2% of all social insects out there that we've cataloged. And it's interesting, though, when you look across the social insects that do have extremely polyandrous queens, these species are highly prolific. So we're thinking about the leafcutter ants, the army ants, where we have millions of individuals per colony, our honeybees, which are some of the most prolific bees we have on the planet, and we know they really are the hero in that sense. And so we can think about whether or not this trait that the queen bears of being extremely polyandrous, technically also called promiscuity. It's not a dirty word here to call the queen promiscuous. She's in fact very promiscuous. We can wonder about what kind of traits this confers, why she's been driven by evolution to have evolved this way. And if we think about just within Apis, across all species of Apis, honeybees, the genus for honeybees, all of them are ubiquitously extremely polyandrous, and to a great extent. For instance, in Apis mellifera, when we have looked across different samples of bees, the range of mates per queen, the range per queen tends to be somewhere between 10 to 20. So the average overall is thought to be about 12. But the record for Apis mellifera is 44 so far, 44 mates per queen, which is huge. If we think about the giant honeybee in Asia, Apis dorsata, they, out of all Apis, are the heavy hitter for extreme polyandry. The average mate number for them is around 60, with a known record of 104 mates per single queen. And these are just the ones we've gone into and looked at carefully. So who knows what they're really doing out there in terms of record-breaking mating on the side of these extremely polyandrous queens. So when we say extreme, we do mean extreme, especially relative to most social insects where the queens mate once, just with one single drone or male. All right, so why are they extremely polyandrous? We know that promiscuity, queen promiscuity, is a risky business for them. They can contract sexually transmitted diseases such as nosema, deformed wing virus. These are probably the best known, well-documented cases of transmission between drones and queens of viruses. And then those viruses can have the potential to be passed on to their colonies as well. This is known in the case of deformed wing virus. So it's risky business. Why do the queens do it? There must be some reason why the risks of this behavior the benefits of this behavior outweigh the risks. And we do know that from a lot of good work that's been done over the last maybe 15 years or so, that colonies that have extremely polyandrous queens, and thus colonies with a lot of genetic diversity in them amongst the worker offspring, they have increased disease resistance, their brood is more viable, they are better able to maintain stable colony temperatures, and the queens that are well-mated, that have big round spermatheca filled with semen, produce a lot more signals as well when they are actually egg-laying. They produce more QMP, and this presumably makes them a more attractive leader for the workers that are hosting those queens in their colonies. So one thing I wanted to know when I got to Tom's lab, he had been looking at the benefits of promiscuity or extreme polyandry in the field of disease resistance. I wanted to know whether or not this kind of genetic diversity improves productivity of the colonies. I really had a sense that these colonies are just profoundly robust in many ways and have the ability to deal with a lot of changing circumstances around them, and I just had a feeling that some of this genetic diversity was linked to this. And if these kinds of productivity benefits are substantial enough that the colonies actually prosper from having a lot of diversity within them, then that would impose, or not impose, it would confer to them fitness benefits, and so this kind of behavior would be selected over the many, many generations that our honeybees of breeding that our honeybees have been exposed to. So when a queen mates multiply, I have alluded to the fact that it introduces a lot of genetic diversity into these colonies, and so most social insects are in what we call the ancestral state. They have a single queen with a single drone male partner, and they produce one family of workers, and we call that a single Patra line. So Patra line is another fancy word for subfamily. And those colonies can be considered somewhat genetically uniform. And if we contrast that to our honeybees, where we have multiple partners for the queen, this is a derived state for social insects. So because most of them don't do it, we presume this is a state that evolved secondarily for them. And these queens produce genetically diverse colonies with multiple Patra lines of workers who are not all full sisters like we would see in a genetically uniform colony. Instead, you get many Patra lines or subfamilies of stepsisters in these colonies. And we do know that these Patra lines differ in their probability of performing different tasks within these colonies. So I don't recommend it, and you know probably better, if you go and kick a colony and then examine the DNA of the stings that you might get for doing such an act, you would find that they tend to come from certain subfamilies within that colony, that some subfamilies are more inclined to be defensive of the colony. If you were to collect up some dead bees, put them into a colony and wait for the workers to drag them out, or by undertaking, pull the dead workers back out of their colony, the workers who take that task up would tend to be from certain select subfamilies within these colonies. And this was evidence that has been gathered throughout the 80s and 90s and really formed the basis of why we think genetics might play a big role in division of labor within colonies. So if the colonies have the potential to harvest up all of these different kinds of genes from the potential drone fathers out there and create multiple Patra lines of workers within their colony, they then have the opportunity to perhaps inject into their colonies a lot of these Patra lines to take care of the diversity of tasks that need to get done in colonies as conditions change. Does that make sense? Okay, I'm not going too fast. All right. Okay, so these queens literally advertise to the drones that are chasing them through the air how to catch them and continue mating with them. This mating sign, this is what's seen in a queen when she returns from her last drone mating into the colony. This is the remainder of the genitalia that's cracked off of the last drone who mated with her. And the workers will remove this mating sign, but in the air, it hangs off of the tip of the abdomen of the queen. and it reflects UV light, and drones are excellent at seeing UV light. They have great UV light receptors in their eyes. They have heads that are wrapped with eyes, if you've taken a close look at your drone heads. And for them, this flying queen at the head of that mating comet has a glowing reminder to them that they need to catch up, chase her, catch her, and then insert their genitalia where they see that glowing UV spot. So she is literally telling the drones, she's signaling to them, continue this chase, continue this flight, here is your moving target. And so I wanted to, as a new postdoc in Tom's lab, think about what some of the colony level effects of this kind of extreme polyandry could be for these workers and their productivity, and also look at worker level mechanisms. So overall, how is a colony more productive? And individually, at the level of the worker, how are those workers from a variety of different Patra lines contributing to a more productive colony? All right, so it's pretty easy, as you know, to manipulate mate number in queens. Thankfully, when you ask this kind of question, you can turn to instrumental insemination and control mate number yourself, rather than hoping for the best as you release queens to the sky and trying to assess in some way how promiscuous they've been before they've come back. So we can create experimentally two groups of colonies. We can create the ancestral form of mating for most social insects, this kind of single Patra line colony where we have a queen that has been inseminated by a single drone. Or we can create a multiple Patra line colony where we take multiple drones from the same drone population and at random, so we don't go through this chase process, we just at random for artificial selection, or insemination, pick or select these drones from the pool of available bachelors and choose them to inseminate our queens. And these queens in these experiments are all super sisters, so they are closely related. The drone pool is the same. What we're really trying to do is create two different types of colonies, where one is the ancestral single Patra line type, and one is the more derived multiple Patra line type, what we would expect to see in a well-mated queen at this point. Okay. And for the very first experiment I did at Cornell, we decided to look at swarming, because swarming is such a tough challenge for honeybees. So if a honeybee colony is going to show its mettle in anything, it's that challenge, especially in Ithaca, New York, of getting through their first swarming season and establishing themselves quickly enough that they can survive their first winter. And this is such a tough task that early data that Tom took when he was in Ithaca showed that most swarms, only about 29% of them survive their first year. The vast majority of them do not collect enough food to survive. And so this is a great test of how productive a colony is under extreme circumstances, and really evolutionarily relevant circumstances too, because this is how our honeybees reproduce at the colony level. So in order to fake a swarming event, it's pretty easy. You cage up some bees. You feed them a lot of sugar, so they're all engorged on sugar like they would be when they leave naturally leave their nest. And then we gave them empty colonies, essentially. And over the next year, we monitored how well they occupied those colonies, how much weight they gained, how much they foraged, how much comb they built. And we monitored them through, I believe we started the experiment in, maybe it was the beginning of June, and we watched them through the whole year, through winter and into the next spring, to see how well they lasted. And these are our two groups. So through the entire presentation, I'm going to keep talking about these two kinds of colonies, the multiple Patra line and the single Patra line colonies. And this is our first test. So what we found was, especially in the first week after these colonies had moved into their new empty homes, we parked, this is Maddie Gerard, she's parked outside of this colony, counting the number of workers as they return to the landing board. And so we did this for 5 days, rotating between colony groups, from colony to colony, counting the number of bees returning per minute. And we found that on 3 of the 5 days, the orange is always the multiple Patra line colony, and the blue, or it looks kind of black from here, is always the single Patra line state. On 3 of these 5 days, the foraging rates were substantially higher, 30 to 55% higher, just a lot more activity of returning foragers to these colonies. And when we tracked the productivity of these colonies over time, so we have some of our first estimates in June, going all the way through winter, down to the following May, and we have our genetically diverse colonies here and our single patriline colonies here, you can see some cool stuff here. First of all, you can see that both sets of colonies are going through weight increases when they're hitting a honey flow, and both colonies are packing on some weight, but our genetically diverse colonies are packing on way more weight when these flows hit, like here and here. Just, they're gaining momentum in a way that those single patriline colonies are not gaining momentum. And information like this is especially relevant because at this point in the experiment, those colonies were the same size. We started everyone with 1 kilo of bees, and this is how they were being relatively productive with the same size population. But over time, these colonies could rear more brood, and they were just snowballing in their ability to be more and more productive over time. Everywhere you see an arrow is a colony death out of the two groups. So you see that colonies died in both groups. We had swarms, unmanaged swarms, that many of them did not survive their first winter. However, for the single patriline colonies, all of them died in an early cluster, either in September or before we even hit the holiday break, basically. So we went from, I think, a group of 10 colonies in this treatment to 0 by the end of the year. None of them survived even halfway through the winter. Whereas the colony deaths in the multiple patriline group were concentrated mostly in the spring, and we still had survivors by the end of the next year. In total, we had 25% of our multiple patriline colonies survive versus 0 winter survival for those colonies that had very low levels of genetic diversity in them. So we're seeing at the large scale colony level that there are substantial productivity differences if colonies have a lot of genetic diversity in them, and this is translating directly into long-term survival for these groups. So that got us thinking about what exactly are the workers doing to create this phenomenon, and that foraging data was the first place we thought about looking. So we started thinking about waggle dancing, so recruitment to foraging sites, a lot of foraging activity. How does genetics allocate workers to the task of foraging? And so what I'm going to talk to you now is going to be about moving into really an analysis of how foraging and recruitment is affected by this promiscuous mating behavior of the queen. All right, so we have pretty similar methods for much of this work, a lot of which you've been hearing about already from me or from Tom. It involves observation hives and a lot of counting and following and tracking individual workers in these nests. So in all cases, we had our observation hives set up. Very often they were paired, so we would have one observation hive that was a multiple patriline colony paired with a single patriline colonies. And in many instances, they were operating simultaneously. So both colony types would be observed at the same time, in the same place, experiencing the same foraging environment. Does that make sense? So everybody's got the same thing, same access to the environment, and they're of the same size and in the same home with similar resources, but the difference is the nature of the population of the bees, whether or not they're genetically diverse or genetically uniform. And then we would set up video cameras, record the dance floor of the colony. So I mentioned yesterday, this is the area of the comb directly adjacent to the entrance of the observation hive. And this is where workers, when they're coming back as foragers with a lot of food, they will deposit the food that they have to a receiver bee, which you heard about this morning. And if they're going to do any recruitment, any waggle dancing, they'll do it at on the dance floor near the entrance. So you can very neatly train a camera on the bottom half of the bottom comb, and you can collect up all of the information visually that the workers are sharing with each other about where they've been and how excited they are about it, basically. Whether or not they dance, where they dance for, and how long they dance to advertise these resources, which is a reflection, really, of the quality of those resources and the bees' experience in general. All right, so we always try to keep the same location. We always did keep the same location, and like I said, the same environment. And then you get into the hard work of just reading these dances, which requires hours and hours. I think I spent in Tom's lab 6 months decoding 4,000 bee dances. I developed a twitch in one eye. It was very difficult, but it was a great way to get a sense of, just the kind of effort that goes into collecting these data. Okay, so let's look at how recruitment compares between these two colony types. We've got our multiple patriline in orange and our single patriline colonies in blue. And we found pretty substantial differences in recruitment. So that was really reflecting what we saw at the entrances, the larger traffic in these genetically diverse colonies. So if the colonies were genetically diverse, overall, the mean number of waggle runs per dancer was 63% longer, which is substantial. And then on a daily basis, these dancers in the multiple patriline colonies were performing more dances overall. So we have more dances being performed over the course of the day, and each individual dance carries more waggle runs in it. So this is an amplification of these important signals that help get bees out there, other bees out there, to the available but ephemerally available food resources that are available. All right, so something that honeybees do is called the shaking signal. Have you heard of the shaking signal? Yes, okay. So in the context of foraging, this is when one excited worker, a forager who has had usually good experience the previous day, a good foraging run the previous day, will very early in the morning move around the dance floor, which is relatively quiet at that time of the day. We're setting up our cameras basically at 7 a.m. And she will move around the dance floor, and she'll put her front legs on quiet workers and shake them up and down. So it's this kind of like up and down vibration, and she'll move around the dance floor, find somebody else, shake them, find somebody else, shake them. And what we found was in these multiple patriline colonies, we had 2 times more shaking signals on these dance floors early in the morning. So essentially, these foragers in genetically diverse colonies were waking their sisters up, their sisters and half-sisters, up earlier in the morning, getting them oriented to the task of foraging. And then once engaged in foraging, they were dancing longer dances and dancing more often. And all of this is good information to understand how that foraging effort could be propelled by a colony of extraordinarily diverse half-sisters of bees. Okay, so what are workers doing to produce these effects? How are they essentially operating to create a higher level of foraging? And there are a variety of different roles that workers can take up as a forager. A worker can be a dancer, and she can also be a recruit. She can be somebody who sits back and waits for information to come to her about what is potentially out there in the world that she should spend her time visiting that day. So these recruits rely on information through a dance from other individuals to get them activated to the task of foraging. But you don't always need to pay attention to dances. There are some workers that are very independent, and as foragers, they tend to pay less attention to waggle dancing signals, and they follow their own internal drivers and operate kind of sometimes outside of the realm of the information that the waggle dance can provide. And these are known as scouts and inspectors. And for both of these, instead of using the external information that recruits rely on, what these guys do is either search for novel resources independent of information from dances. The scouts like to go out and find new things, and they play an important role in the colony, not only in the context of swarming, but for foraging too. It's their job to find the next best thing that's out there, or not the next best, the next new thing that's out there, and move beyond the resources that the colony already knows about as a collective through the dances that they're sharing. Or you could be an inspector who has the propensity to go back to foraging resources that you knew about from your previous work and check out and see whether or not they've become available once more. And you can imagine this kind of behavior would be really useful in the context of foraging on flowers that bloom for a limited amount of time during the day, or have just become available and were productive, and you want to make sure the colony is capitalizing on them for as long as possible, as long as those resources are available. These guys tend to become active foragers without the information that comes from recruitment. So we wanted to break down all these different types of ways workers can get engaged in the foraging effort and see how genetic diversity affected those. Okay, so our general methods were things you've been hearing about already from both Tom and I. Lots of times we train the bees to use feeders. So we had known food resources that were standardized across the bees. So this is important because the nature of the dance can change depending on the quality of the food that the bees are visiting. So we want them to all be visiting, having basically the same experience at the feeder. Same quality of food, same distance to it, all of those things that go into their assessment of that food source. So we train the workers to feeders. We monitor them in observation hives. And then we specifically look at the dance floor, tapes of the dance floor. Can you see these workers here that have all the painted, that's not just for fun, that's us at the feeder when a bee becomes trained to that feeder, marking her with an individual paint code that tells us that she is red, green, or blue, yellow. And this is always fun for the students who like to come up with names like she's Brazil or she's Christmas or something like that. But everybody has to have their own unique name that lets us know who they are as they're working with this feeder. And then and this is the hard part, especially Tom mentioned how you become sort of attached to these bees as you watch them work hard all day. At the end of their foraging career, we have to sacrifice them early and then get a sense of their DNA, extract their DNA so we can identify who their drone father is and figure out how their activity level relates back to the parent that they came from on the male side. Does that make sense? Okay, great. Okay, so this is a shot of one of our feeders in the same glass house that Tom showed you this morning at Cornell. And here's our observation hive down at the other end of the room. And we, again, put these feeders into these situations so that we can tightly control the experience of the workers and prevent them from going to other food resources. Okay, so let me walk you through this figure. This is a typical multiple patraline colony. We tested several of them, and we also tested several single patraline colonies as well. So what we did is we looked at all of the feeder visitation and all of the dancing that was done for that feeder within the greenhouse for this colony over the course of 5 days, added it all up, figured out which father these workers came from, and then divvied out the total amount of work that was done in terms of the group foraging effort to the 14 patralines that were in this colony. Now, we did intend to have 15, but we could never find evidence of the 15th in this colony. So for whatever reason, the insemination resulted only in there being 14 patralines in this group. But the important point is it was really the top 3 dancing and feeder visiting patralines that accounted for the vast majority of the work that was done. Up at the top here we have total time spent dancing from all of the videotapes we took of that observation hive dance floor, and the total number of feeder visits that were made at the other end of the greenhouse for the person, me, who was hovering over this feeder, writing down the identity of every bee that showed up at that feeder. And so these top 3 patralines in this colony each accounted for over half of all of the activity that was done, the foraging and recruitment activity that was done in these colonies. And the range here shows across all of the multiple patraline colonies that we looked at how much of this activity was accounted for by the top 3 patralines. So we tended to have 3 specialist patralines when it came to feeder visitation and recruitment in these colonies. That was true. Here's some other histograms so you can see how that's somewhat similar across. And what we found was that 20 to 57% of the foragers in these single patraline colonies never danced. So for instance, when we gave them the best sugar we could give them, best sugar water, we had one colony that even though they visited this feeder consistently, it wasn't with gusto, but they did go back and forth and visit this really rich feeder over and over again, we couldn't find a single dance on the dance floor to report to anyone else back home that that feeder was present. So how dysfunctional is that the opportunity to visit this very rich resource was never reported back home, and that's what kept the forager rate really low in that group. So here is Maddie again by her observation hive. This is how we spent our time for about 6 weeks one summer. Her at the observation hive, me hovering over the dish. And she wanted to, for her senior thesis, she did this during the summer between her junior and senior year. She wanted to do something with all of the data that we collected. And so we decided that she would go back into the videotapes, pick focal dances at 2 of the sugar concentrations that we looked at, and count the number of bees that followed those dances and for how long they followed them to get a sense of both sides of the equation of recruitment. You have to have dancers who want to share information and dance followers who want to receive that information. If you only have one side or the other, you don't have any recruitment. Recruitment is a combination of those 2 things happening together. So for her thesis, she dove back into the tapes and extracted some more data. And what we saw was from these dance floor videotapes, where we've got our cluster of marked bees on the dance floor, if we looked at the mean number of followers per dance, this was consistently higher in the multiple patraline colonies than it was in the single patraline colonies. And not only was the mean number of followers per dance higher, so was the mean number of circuits that they followed. So you have bees that are dancing, the foragers who are dancing more often, they're dancing for longer, and you have a larger audience of dance followers for those longer, more frequent dancers. More individuals following individual dances and following them for longer as well. Pretty remarkable, I think. All the way around, we were finding amplification of the signal sharing for this really important trait that our honeybees do of waggle dancing and sharing information about food resources. All right, so this is Morgan Carmichael, and she was the first thesis student I had after I left Cornell, stopped working with Maddie and Tom, arrived at Wellesley and started having my own students. This is Morgan here. She's now doing a PhD with Marla Spivak, so she fell in love with honeybees at first sight and is continuing a career in research with them. And this is her latex-covered hand right here with an open dance floor, and she's holding a very special kind of pen. And this pen, I hope you can see it. I can barely see it from where I'm standing. There should be a long, skinny thing coming out of this pen. Can you guys see that? So at the very end of this pen, it's really a syringe that you can eject a very flexible fiber from, and this fiber soaks up the smells that are being produced by living things. And in this case, we wanted to get a sense of not only what was being produced in terms of smell in the colony, but also on the workers' backs. And that's because workers, when they are waggle dancing, produce a set of 4 pheromones that are known really only from this foraging context. And there are 4 chemicals that radiate off of these hot workers as they're dancing. And they're known to, if you can get synthetic versions of these chemicals, and if you inject them into a colony, they actually increase the rate of forager exit from that colony. So they're thought to be stimulating pheromones or chemicals that are another form of signal that enhance the message of the waggle dance, which is basically get out there and find this food. So one thing we did, this was Morgan's thesis, so I'll just tell you quickly one part of it. One thing we did is we took these flexible fibers, put them on the dance floor of the colony, exposed them for 30 minutes, and looked at the total number of these 4 chemicals that we could collect from the colony over this period of time. The more chemicals they are, the more they get soaked up on this fiber. And what she found was that in multiple Patra colonies, this chemical signal was 12 times stronger in colonies that had multiple Patra lines of workers compared to the single Patra lines of workers. And these were equal-sized colonies again. So same number of workers, just a lot more signal sharing, not just the physical waggle dance itself, but also this chemical signal that goes along with waggle dancing. So next we moved on to thinking about inspecting. On our quest to think about all of the ways that signal sharing could be improved by multiple Patra lines in a colony, the promiscuous behavior of these well-mated queens. And so we looked at what inspectors contribute next. And we did this with a study. This was at Cranberry Lake, which Tom introduced you to this morning if you were here for this morning's lecture. It's an environment where you can take a colony. There's really no other bees up there, not good forage. It's all water and coniferous trees for the most part. And you can put an observation hive in this environment, put out feeders, and the bees will go to those feeders because they basically have nowhere else to go. One time we discovered a hummingbird feeder that the bees could barely get their tongues into, and there was a crowd of about 100 around them. They were so desperate for food in this area that once we took that hummingbird feeder down, we felt pretty confident that we were the only game in town when we put our feeders out. So we, again, have pairs of single Patra line and multiple Patra line colonies, same environment, tested at the same time, side by side, same size, sister queens. The only difference is the level of genetic diversity within these 2 groups. And we trained each of them to their own feeder, one 300 meters in about this direction and one in 300 meters in the opposite direction. And what we wanted to do was get the colonies used to using these feeders, take the feeders away. Well, we didn't really take them away, we just emptied them. We put the nice sugar water that we were providing them in a jug, sealed it up, washed everything out, put the feeder back together, so it was now the equivalent of an empty flower, a flower that had been offering food reliably before then. We emptied that food, and then we looked at rates of inspection between these 2 colony types, and then filled those feeders back up again after an hour to get a sense of how much they inspected those empty food sites when we did empty them, and how quick they were at harnessing, rallying the troops basically, getting everyone back there once the food was once again available. And we compared that between our 2 colony types. So tracking feeder visitation and recruitment over what we called the hour for inspection and the hour for foraging. Good? Make sense? Okay. So, surprise, surprise. When we look at the multiple patrine colonies, first we looked at the total number of feeder visits during this hour. So these are visits to empty glasses, basically, that no longer contain our rich sugar solution. And the bees are, showing up, keep showing up, but there isn't food yet available in them. And the rate of feeder visitation during that inspection hour was about 76% higher during this period. Not only did we get more rate of feeder visitation per bee that acted as an inspector, there were more inspectors in general in these colonies that were from multiple patrine colonies, or these workers that were from multiple patrine colonies. And so when we replugged that feeder with new sugar syrup and the food was once again available, the response was very swift in these multiple patrine colonies because of their regular, consistent check of their inspection of these sites. They were able to recruit very quickly their workers back to their feeder in comparison to the single patrine colonies that were not quite as swift at this task. So we're not only looking at feeder visitation at the feeders, we're also, of course, we've got students back at the colonies videotaping the dance floors, pointing at workers that are dancing, and then we can estimate dance recruitment as well through these tapes later on. And when we did that, the mean number of dances over all of the reps that we did for the multiple patrine was close to, the multiple patrine colonies was close to about 5,000 compared to 900 waggle runs for those colonies, same number of colonies that only had single patrines of workers within them, so not quite as well mated. What was really interesting was what happened with the bees that were not marked. These recruits that once the feeders became available once again, you can understand during training, every bee that shows up to that feeder, we consider her to have learned about the feeder, and we mark her, we give her a unique color code. But if we have any bees show up to this hour of foraging after the hour inspection, and they don't have a mark on them, that means that they have just discovered that feeder. They are newly recruited to that feeder, and they have done so by following the waggle dances back home. And we saw 4 times more recruits brought to these feeders that were from colonies that had a lot of genetic diversity in them versus the single patrine colonies. So I just want to end talking about how scouts enhance the foraging effort. So recall, scouts are those bees that go out into the environment. They are looking for, they're novelty seekers, we call them. They like to look for new things, new shiny things. They're the reporters of the hive. They are finding the newest thing out there to report back to everybody else. And again, we set up pairs of colonies. To get bees to scout is pretty easy. You take them from a familiar habitat and you move them to an unfamiliar habitat where they don't know anything. The recruits will not go out to find food and come back and dance until the scouts have done their initial work of discovering all of the food in this novel environment. And one of the things we did after we recorded these dance floors was look at the number of scouts in a colony and allocated them amongst different patrines. And what we discovered was that colonies could have a range of these specialized scouting patrines, or what we called scout-rich patrines, where the scouts were concentrated in those top number of patrines like we saw with our greenhouse experiment and the feeder. So if we look at the number of scout-rich patrines in a colony, one of our colonies, our multiple patrine colonies, actually I think this might be 2 of our, no, because we only had 6 in total. So this is one block here. One colony we couldn't really identify a clear scout-rich patrine. But the rest of them had at least 1 scout-rich patrine or 2, and many of them had 3, just like we saw with the previous feeder data, that multiple patrines of workers who were really invested heavily in this scouting activity. And what we found was the number of dances that we could count in a colony over the course of their first day in this novel environment was really directly correlated with the number of scout-rich patrines these colonies had as well. And I have to say, with biological data, it's always messy, and to find a significant correlation with only 6 data points means that you have a pretty good pattern happening here. And not only that, by the end of their first day, the colonies that had multiple patrines of workers in them had a much farther foraging reach. When we decoded the dances that they were doing, they were 3 kilometers farther out on average in the course of that day. And you could see the radius of the colony expanding over the course of the day as they fan their way out over the environment. And by the end of that first day, they were 3 kilometers farther in their new environment if they were from a multiple patriline colony. So we can say that the promiscuous mating behavior of queens who have the opportunity to mate well and mate with many good males in their environment, are helping their colonies in multiple ways when it comes to developing a really rich, robust, and productive foraging effort. We're getting more dancing foragers, a lot more attention paid to those dances, and this would include both brand new foragers and those that have been foraging for a while but consistently like to look to waggle dances for information about where to go. But we're also seeing the forager categories where they don't pay as much attention to waggle dancing, these inspectors and scouts. They're also helping to amplify the signaling, the pool of signals available to everyone if they're from a colony that is genetically diverse with a well-mated queen. So this summarizes basically what I just said, but it explains some of the reasons why beekeepers should be really interested in understanding how well mated their queens are. And I know that this is not easy to do. You don't have access to the genotyping facilities that researchers have, and so there's no easy way for you to assess the genetic diversity of your colonies. I can say, though, in the US, and I'm not sure what the case is for the UK, that we have had, both in the 1990s and in the last 10 years, good estimates made by researchers where they blindly order without identifying queens from different breeders around the area and then take those queens and make an assessment of the queen quality, looking both at how full the spermatheca is, and in the latest survey done by Dave Tarpey at North Carolina State University, looking also at the genetic diversity of the sperm that they carry. And so there is an interest in this. It has to be, to be well assessed scientifically. It has to be a rigorous scientific effort. But I think it is a good way to check in on the status and the health of your queens, knowing all of this information about how important it is that they be well mated to produce really productive colonies. So I have a ton of people to thank at Wellesley and at Cornell. I specifically want to thank Tom Seeley, who we've all thanked for being here today and the last couple of days. But he was such a wonderful host at Cornell, and all of this work, I learned so much from him, following him around and painting bees and chasing bees over the Cornell campus. Thank you for your patience, and I'd be happy to take questions.
With the experiment where you showed that most of the foraging was done by the top 3 Patra lines.
Yes.
How did that reflect on the overall representation of the Patra lines in the colony? Were they the most frequent Patra lines, or were they selected specially? Do you follow what I mean?
Yeah, so did they seem to be the most active because they were so well represented in the colony?
So it was based on numbers, really?
Yeah, so when we look at the breakdown of the actual distribution of number of workers from those colony populations reflected as, it was not what you would have predicted. So there was an over contribution of activity from those workers within those Patra lines. I see what you're saying. We have to compare the number of workers in each of those Patra lines to their general contribution to the activity.
Do they favour, when recruiting for a job, their own full sisters? Oh. Do you follow what I mean?
Do you mean, do they like to dance for their full sisters versus their...
Yes, I mean, they recognise their full sisters. So when you're recruiting for a task, is there a greater tendency to recruit a full sister than a half-sister? It's possibly an impossible question.
I don't think it is. There isn't extraordinarily strong evidence that they favor their full sisters over half-sisters when they're doing things within the colony, whether it's swarming or sharing information. There's actually not very convincing evidence that happens. And so if you imagine the environment that these workers are dancing in naturally, for us, what we do is we take the sides off of these glass observation hives, we fill them full of light, and we watch them as they dance in this glass-covered environment. But it's very different than what they are doing naturally, in the dark, with vibrations and smells, jostling around with everyone. So we really have no indication, although it hasn't been tested by me. we have no indication that they are favoring doing dances for their full sisters. I also think it would be kind of hard to get that done. If you've ever watched a worker dancing in an observation hive, and I'm sure you have, they'll do a little bit of dances down here, and they'll stop, they'll go up here, dance a little bit, they'll go down here, dance a little bit. So they are moving around actively. And even while we track the dancing, the following that the workers are doing for these signals, they are getting bumped and jostled around and pushed out of the way all the time too. So I suspect it's far too messy an environment to start choosing your favorites.
Pretty compelling evidence, really interesting. We can see that there is clearly a correlation there. This increase in activity is related to diversity in genetics, that's fine. Mm-hmm. The question really is, why? Because each of these individual patrilines, if they were in a single patriline colony, would presumably perform as if it were less active. So what is it about the mixture of diversity that makes that level of activity rise? Yep. And secondly, is there a point at which that happens? So would it be 2, 3, 4? Uh-huh. Or if you hit 7 or 8, does it then start to rise, or is it a logarithmic type scale? Or
yeah, good questions. I can say from my own research, when I first got to Wellesley, one of the things we did was we independently tested many single patriline colonies on their own in this greenhouse environment, all super sisters, same kind of setup, but serially tested them in a greenhouse at Wellesley. And then we made a super colony out of them. We gave them a brand new queen that was another super sister, and took all of these 15 separately tested patrilines and made one big multiple patriline colony out of them. And the results were really interesting. We only did it once, so we only have one test for a multiple patriline colony. But what we found was many of the single patriline colonies on their own were duds. And this was the first summer my students were working in the lab. They found the work quite boring because initially it was very, very slow to look at these single patriline colonies as they utilized a feeder weekly in this observation, or in this greenhouse, until they got to what we call the super dancy colony, which was a single patriline colony. And all of a sudden, on everybody's texts started going off because the student who was monitoring the feeder could not keep up with the level of, basically the feeder was attacked by this really strong reaction of this single patriline colony, the only one out of the 15 that we tested separately. And then when we put everything together, we haven't completely analyzed all aspects of the mega colony that we created, but it was nowhere near as active as this one super patriline, this one super dancy patriline that was tested separately. So, okay, all that to say is, I think when you end up getting a diverse colony together, you have multiple patrilines that are good at different things. So, for instance, often when I present this work, beekeepers will say to me, I hope you saved the genotype or the DNA of that really great colony so we can start getting that kind of genetic information into our bee breeding systems. And really, I don't think that's the way this works. I think you have some colonies that might look like duds when it comes to a feeder test in a greenhouse, but they are probably doing other things really well. And together they are doing that magic thing that evolution has done for us and put together these highly productive colonies by taking a little bit of all of the potential genes out there and having the queen have this good opportunity to wrap them up into a single colony. I can say, let me think to answer your other question. Dave Tarpey, who also worked in Tom Seeley's lab, he was a postdoc before I was there and is similarly interested in this idea of genetic diversity and has mostly looked at it with respect to mating number and disease levels. He did a really interesting study with colleagues in the US where they looked at commercial beekeeping operations and tried to get a sense of how much honey colonies were producing and the level of genetic diversity within those colonies. And the magic number, at least in their sample that they found, was at about 7 mates per queen. That's when you started to see real differences in statistical differences in quality. So that's the best information I can give you for number 2. Yeah.
Did you find when you looked at the scouts
at the what?
The scouts and the inspector bees, were they from the same line, and also would they then follow on and become the bees that would then coordinate swarming, etc.?
Oh, good question. So when we looked in the colonies for our inspector bee experiment and the scout bee experiment, we did find an effect where we understood we took 3 populations of workers to compare. We looked at the general colony population, the general forager population collected as workers returning to the entrance who were coming back with food, so just generalized foragers not knowing their roles, and then also specifically collected those inspectors and scouts. And we found that the foragers, the structure of the genetic population of foragers generally differed from the broader colony population, but interestingly, the structure of the inspectors and the scouts also was significantly different from the general forager population. So there were standout patrolines within those forager patrolines that seemed to be a sort of subset of that kind of activity. I think, did I answer all your questions? There was one more, I feel like. Oh, swarming, yeah. So the most we have done with swarming was a test really where we set up swarms side by side and allowed them to go through the house hunting process side by side and then present these data. But when they had that opportunity to, race, they didn't know they were racing each other, but they were in fact, I thought they were racing, diverse versus a uniform colony side by side on a house hunt, and did this over multiple iterations. What was interesting was the uniform colonies tended to consider as many options, but they did it slower, and they took longer to take off. Whereas the multiple patrilineal colonies they basically collected the same number of opinions, danced the same number for whether or not, when they would before they finally made a decision, but they came to the decision faster and shared information faster. We didn't, in those cases, specifically collect the scouts to identify them. So all we have for that study is colony level stuff. We don't know how it breaks down at the level of the worker or patriline. But we do know there's an effect in swarms.
What advice would you give to a small hobby beekeeper who wants to benefit from the research that you have done?
Yeah. I think I outlined at the end how difficult it is for hobbyists or commercial beekeepers to really get a sense of the genetic diversity of their colonies, but I will have you think back to Kim Flodham's talk from yesterday where he spoke about getting a sense of who's in your neighborhood, how many bees are in your neighborhood, how many colonies, and who's potentially contributing to your gene pool. So I think that is, entry-level investigative work for a hobbyist beekeeper, is to get a sense of who their neighbors are, how healthy those colonies are, and the kind of potential dating scene that your queens are facing, basically, when they're out there in these DCAs.
With regards to the sequence of insemination, was there any correlation? Was there any correlation to the primary 3 foraging groups that you found?
Oh, got it. We didn't know the order of the collection of semen. Basically, we had queen breeders do this, so it was done blindly for us. When we were testing these bees, we didn't know whether they were genetically diverse or uniform. It wasn't until we finished the experiment, we would open the envelope that the queen breeder provided us with, and it would say this group was diverse and this group was uniform. But the way that they would create the queens for us was to harvest the semen, mix it in a dish with a glass rod, and then do the insemination. So our best guess is it should have all gone in there relatively uniformly. When we do the genotyping later, we get a sense about whether or not that works, and it tended to work well. We tended to get what we expected. What else can I say? Well, when other people have specifically looked at queen insemination and how that reflects the kinds of bees that are produced, it turns out that this rule we have in animals when it comes to mating and the evolution of mating systems, the saying is the last one in is the first one out, meaning the last male to inseminate a female tends to be the father of the majority of the offspring. But when it comes to honeybees, that's not exactly how it works. We see some of these effects, sort of uneven fertilization of eggs very early on in the queen's life, but within weeks to a month or so, it's a pretty good mix that's gone on in that spermatheca, and the eggs are being fertilized fairly evenly across the board.
Yes, you make a strong argument for diversity, and yet you say that only 30 of 40,000 species of insect do this. Have you got some ideas about why that might be such a low ratio?
Yeah, that's a really good question. And it's a long one to answer, but I'll do my best here. The thought is that the level of relatedness within these colonies tends to drop off markedly when you start to introduce a whole lot of drones into the mix. And so you go from families of tightly related individuals where everybody shares a lot of genes and the same genetic interest. With diversity, you get a situation where the queens have created colonies where, in fact, the level of relatedness amongst these colony members is actually quite low. Can I see you back there? Yeah, it's actually quite low. And so we would expect overall that colonies of social insects tend to favor this high level of relatedness. So the baseline state that we would expect from social insects is that the queens would produce this really tightly knit, genetically closely related group of super sister offspring. In a couple of instances where the queens have gained very tight chemical control over reproduction of their offspring, where the workers really have little choice but to be sterile workers, we see the queens monopolize or taking advantage of this situation where they do. Not all social insects have great chemical control over reproduction like the queens in our colonies do. Most of them have to bully their way through this effort. They have to police their daughters, eat their eggs, and try and stop them from cheating in the system. So where you have systems where the queens have this kind of chemical command of their offspring, you tend to see a lot more of this kind of multiple mating evolve. That helps to explain it.
Sorry. For an observation then for hobby beekeepers, aside from trying to know if you're getting bees, where the bees have come from obviously, and if you can, the history of the queen or where you're getting the queen from, because I've had a hive that I saw the queen, it was a swarm, it was doing okay, but then when I opened the hive, it was really going quite small compared to other bees that I have, other hives I have next to it.
Mm-hmm. So you mean the number of bees was not that great?
Both the colony hadn't expanded like the other ones had, and the queen herself was very small. She had a very small abdomen. My assumption was she hadn't mated very well. And then to me, that's still the observation. I've got 3 hives, but I'm worried they're about to swarm while I'm away, actually, and this hive is still going not backwards, but very small. So would that be something for advice that you can have? Would you see that in a queen physically as well as your observation of the hive? And
No.
the only other thing you can do is know the history of the queen or not?
I think the last thing I would want to do is start recommending that if you think your queen is tiny, to start chopping her head off. So the best way to tell without genotyping is to dissect out the spermatheca and take a look at what it looks like. A nice plump spermatheca, but that involves destructive sampling, so it's not something you can do either. So in your scenario, just getting to know your bees better, don't start suspecting small queens of anything, just assess them through other things. Like one of the ways that it's not a foolproof way, but your queen should be attracting the attention of her workers, and a lot of her ability to keep her workers happy and attracted to her has to do with how large her spermatheca is and how that's affecting her ability to produce pheromones. So I think more than anything, you should pay attention to the interest of your workers in your queen rather than trying to make external assessments about her size.
That's okay. She's still quite happy and quite alive, trust me. Thanks.
Good.
Quick question again about advice for hobby beekeepers and your thoughts about the practice of drone culling as a method of controlling varroa.
Yeah, I thought that Kim did a good job yesterday of saying the costs and benefits of that. That it can be an effective way, if you didn't hear the talk yesterday, of trapping some of your varroa into a group of useless bees that otherwise take, energy from the colony, and moving those frames out, killing that brood, and then in case of Kim, feeding it to your chickens. The other side of that story is you are taking out the reproductives from your colony. And colonies produce drones when they are really strong. And so a colony that's producing lots of drones is pretty strong relative to its neighbors that might not be producing many drones. So, I think that is a double-edged sword. And I think maybe one of the best ways to try and address it, and I haven't done it personally, is to try and do a bit of both. Maybe provide, if you are a person who is trying to mate your queens locally or you're working with people who are trying, would be to favor having the drone brood in the colony during the mating season when you're actively mating your queens, and then trying some of these culling measures later on or beforehand, but that's all restricted by the length of time you have access to a good foraging season. You know, in the US and Canada, that can be more and more restricted as you go north. Somebody told me yesterday that they had bees flying and rearing brood all year round. So maybe, depending on where you are, you could try and hedge your bets by doing both a little bit when either is possible. But other than that, there's no perfect way to get around that puzzle.
Heather, thank you very, very much.
Thank you, everyone.
Thank you.